I'm really super happy about being here, and so I thank you for the invitation. It's particularly nice to be invited by computational neurobiologists, since I am a completely experimental biologist. And as it was just said, I'm actually very interested in science education, so it's very nice to be on an educational program here. So as a downside, I really ask you to ask questions. So I make a deal now that if you will not ask me a question while I'm having my talk, at the end of the talk, I will ask you questions. So just interrupt me anytime. As you will see, the work I'm presenting is a bit kind of multidisciplinary, and so maybe as some extent hard to someone and not to others. So, autism spectrum disorders, you already heard a little bit in the previous talk, characterized by those features that are social deficits. Social deficits includes many things, including like eye-to-eye contact, but also verbal and non-verbal communication, like our hands. I'm using them a lot. And also kids with autism have the presence of repetitive behaviors, and so they are kind of inflexible eventually to adapt to new routines. Now, unfortunately, those core features of autism, they go readily alone, but in fact, most often coexist with a number of other problems, including intellectual disability. So about 50% of those kids will have, to some extent, some intellectual disability. Motor problems are very common. So for all of those that are interested in cerebellum, please look at the cerebellum in the context of autism spectrum disorders, because it's absolutely a very interesting and new topic coming up. Anxiety, again, a very interesting part. We don't know anything about what anxiety in autism is. In fact, we don't even know whether anxiety in autism is the same as anxiety in other individuals or type of populations. And then finally, epilepsy. About 25% of individuals with autism have also seizures or repetitive seizures. So how do we study those disorders? So we really start from the genetics. We are looking into the genes that eventually are mutated in those patients. And once we have a sufficient amount of evidences, we transfer this knowledge to animal models. So we build what we call construct valid and eventually face valid animal models, such mice. And we also study, and I will mention those later, 2D and 3D cellular models to really understand a little bit more or fill the gap between mouse and humans. So I mentioned our approach is quite multidisciplinary, and this is because it just is the nature of the type of work we are doing. We're asking the molecular basis and the underlying developmental issues of autism spectrum disorders. And so we have really to employ a number of different techniques. So we employ biochemistry, molecular biology, imaging, electrophysiology, and behavior. And we try really to go all the way through. And you can imagine here how important it is that, in fact, you're not compartmentalized just in electrophysiology and behavior versus molecular, but we are really trying to unify the gaps. Now, as one thing that I mentioned before in one of my questions, we know that autism spectrum disorders are just not autism. So we typically think about autism as one thing, but in fact is more like many different things. That's why we call them autism spectrum disorders. So it's a group of disorders of different variability, but also with different underlying causes. And when I say this, in this case, I refer to the genetics, so that we have a number of different genes. And so far, we have about, sorry, here is very low, but about 120 genes with what I call here score 1, meaning that those genes that are really highly associated or highly penetrantly, so when are mutated, they really lead eventually to one of those disorders. But overall, we have well over 300 genes underlying autism spectrum disorders. Now, typically, what we do, not to, not because really the biology requires that, but because we are a little bit, so it becomes handy to us, we try to group those genes, so those genetic mutations that we find in those patients in groups. And this really just helps us to reduce a little bit the complexity of those disorders. And so one of those classes of genes that have been associated with autism classically are those that are involved in synaptic development and plasticity. So those are really, in fact, until a few years ago, we referred to autism spectrum disorders like synaptopathies, and implying that genes related and protein related to synaptic functions were eventually mutating in autism. And this is certainly true, but in fact, what emerged in the last decade from the beginning of the new era of genetic sequencing is that, in fact, a number of other proteins and genes can be associated with those disorders. So some of them, for instance, are those transcriptional regulators, or it includes chromatin remodelers, as well as histone modifiers, or, for instance, genes and proteins related to the proteostasis, so the homeostasis of protein in a cell. So how much protein is either produced or is degraded in the cell. Now, in order to really give you a little bit of a feeling about what we are doing today, I decided to present you one kind of longer story and then a few slides in another one. And the first one is really on the connection between the transcriptional regulation and synaptic development. So what we are doing, so as I say, so the first part is going to be between genomic and synaptic plasticity. And something like genomic plasticity is probably something that not everyone in this audience is used to, but it kind of refers to the possibility and the capability of our genome or the chromatin to undergo a number of different modifications that makes the chromatin more or less accessible, and so that makes it more prone to transcription and so gene expression or not. And so there are several enzymes that are either called, for instance, for histone modification, they're called writers or erasers that add modification or remove modification on the in this case, an histone level to make the chromatin more or less open and so more or less in a state for prone to gene transcription. So over the last year, I became very fascinated by the fact that as neurobiologists, we always focus about what happens here. So what happens at the synapse, and certainly, it's very important what happens there, right? So anything that whenever there is an input in the central nervous system, eventually there is a reshaping of the synapse. And this is very important to kind of reshape the neuronal network in an experience-dependent manner. But we often forget about the other side of the cell, is sometimes far away, but still is there. That is the nucleus. And so, as I just mentioned, the nucleus also undergoes a number of different modifications. And so that's what I call here genomic plasticity. And what I'm trying to going here to is that those two things eventually are communicating and are important for each other. And so how to connect that with autism spectrum disorders? I mentioned that we know that chromatin modifiers and histone modifiers are often mutated in autism spectrum disorders. And so what we did for this study, we select 4 of those genes that when we are mutated in all of them, so like each separately, eventually they are connected to autism spectrum disorders. And when I say, for instance, 35 is about 0.7% of patients with autism and or intellectual disability have mutation in this gene, and pretty much H1L follows, and those two are a little bit rarer. So what did we do here? So we wanted to understand whether those genes or the deletions of those genes is somehow connected with a change in synaptic plasticity. And to do this in a sort of systematic manner, we went on looking at 2 different forms of plasticity. And here, I'm sure I don't have to explain much of that because probably you are even more familiar than me about that. So one form of plasticity is the Hebbian plasticity. So I say here, learning. Right, so this is the form of plasticity where single synapses that are used or independently of what else happens in the neurons, they are either reinforced or suppressed. And so this is the very classical form of plasticity that we all of us study. The other form of plasticity is actually the homeostatic plasticity. And this has to do with the fact that if you go over and over through those kind of cycle of reinforcement of a synapse, for instance, you would eventually the synapse would eventually end up in a range where it's not dynamic anymore. So where basically anything cannot happen anymore because eventually there are, for instance, too many receptors. And so neurons undergo this form of plasticity to kind of reset the synapses in a level, in a range where those can be still functional. So basically, it's like imagine that the good dynamic range for a synapse to work is here. Eventually, if we are stimulating a lot of neuron, eventually the neuron will end up on a synapse at that range where it cannot move back and forth anymore. And so the homeostatic plasticity makes it possible to go back to the functional dynamic range. And so the difference between this and this is actually that this, so this homeostatic, sorry, there is a typo there, but homeostatic plasticity or scaling up or down is something that happens over the entire neuron. And this is important because then the ratio of the strength of the different synapses input in the neuron is maintained, because this is important because it reflects the inputs, or basically reflect what happened during Hebbian plasticity. So two different forms of plasticity, although one has to be careful because eventually homeostatic plasticity and Hebbian plasticity, they are also connected, but they are considered two different forms of plasticity. So how to address this in a sort of systematic manner? Well, so what we did is constructing 4 different, or actually 8 different animal models. So on one side, for each of the genes we chose, we did a constitutive plus minus animals. I forgot to say, I'm sorry, that patients that have mutations in those genes, they have heterozygous or de novo loss of function mutations. So here, that's what we are modeling. We are modeling heterozygous mutations. And then we also built a stop heterozygous mutation, a mutant that I will refer to later, I will explain later. And so there we study synaptic plasticity, so both Hebbian and homeostatic plasticity, but of course, because we are interested in what happens in the nucleus and because those genes are relevant to the nuclear functions, what we did is also study the genomic plasticity. So what happens in the nucleus when eventually the mouse is undergoing Hebbian, and the brain is undergoing Hebbian and homeostatic plasticity. So there we perform RNA sequencing and ATAC sequencing. Now, one problem that there is always in the field, and I'm not saying here that we are solving it, but we are trying to, again, to fill a little bit the gap, is about, is mouse at all a good model for autism spectrum disorders? I mean, we are modeling features that are highly human, right? Like sociability and flexibility. And so is that a good model? So, well, it is a model. It's one of the things we are available. Another one that we have available are what we call human cerebral organoids. So in order to really understand whether what we will observe eventually mouse is kind of relevant also for the new human cortical circuits, what we also did is generating corticals or human cerebral organoids. So those are just three-dimensional structure resembling, so they are not mini brains, they are just a three-dimensional structure with cortical cells that are sort of organized, certainly not in the nice layers that we have seen previously. But yet organized, and so we for the same genes we generated cerebral organoids starting from human cells, and there study homeostatic plasticity. Obviously, we cannot study Hebbian plasticity in organoids as is associated with learning, and the genomic plasticity. So what did we find? So starting from the mouse and from the erbium plasticity, so we assess that at the behavioral level as well as the electrophysiology level. So at the behavioral level, we did a classical in neurobiology that is context sphere conditioning. So the animal is placed in a chamber with certain visual and olfactory cues. And in that chamber is actually received this 4 times a current shock. So then now the animal should make the association between the context and the negative experience. And so when we place the animal back in the same chamber, so after the training, the animal goes to the home cage. And when we place it back in the same cage, when I experience the aversive experience, then here we should sort of recall that that was not a good environment where to be. And so the animals would eventually, mice, the way, and sometimes we do that as well, the way we're showing, they're showing is by freezing. So it's a new context, and so it's a bad context, so I freeze. And so by counting the number of freezing, we can kind of assess how well the animal remember and the association between the negative experience and the context. And so what we see here, I'm showing the results for 2 of those mutant animals. What you can see is that the animals actually, so they are placed here in this bad environment, let's say, where they will receive shocks. We let them adapt, and then here we start delivering the current shock. And you can see that the animals basically start freezing more and more. So really indicating that they are learning or they're understanding that something bad is happening there. The curve between the wild type and the mutant look quite similar. In this other genotype, we have a little difference. So it seems that they're a little bit worse. It could be anything, could be related to some other type of phenotypes, including some pain perception, but still we see that here we have a learning curve. However, when we put the animals back in the same chamber 24 hours later, both the genotypes show a significant reduction of the freezing, indicating eventually an impairment in the learning process or the memory retention. So the electrophysiology correlate of this experiment is measuring long-term potentiation. And so that's what we did here. We record long-term potentiation, so stimulating the Schaffer collaterals and recording from the CA1. And so what happens in the wild type one after stimulation is that, so this is high frequency stimulation, that we have an increase of the excitatory postsynaptic currents that we're able to record due to the fact that the synapses get potentiated, right? So they are more receptor eventually inserted. And so that's why we see this higher response to in our recordings. This type of this LTP then is completely gone in the 2 different genotypes, so in our H1L and KDM6B mutants. The dynamic of it is slightly different, and I think this is not unimportant because it gives also the possibility to show that not everything is the same. And so, for instance, here you see that even the early phases of LTPs are affected, while in the KDM6B, this is not true. So just the later phase of LTP is affected. So this is all about erbium plasticity. What about the other form of plasticity we are looking at? So this is homeostatic plasticity. So one way to study homeostatic plasticity in vivo is by injecting in one eye of the mouse TTX, which will block practically any sodium channel and so activity. What happens is that this eye is projecting to a visual cortex. And the very nice part of this experiment is that within one animal, you have both the control and the experimental, the treated one. So because in fact, the treated eye project one visual cortex while the non-treated eye to the other visual cortex. So what you expect here is that practically by killing the input to the visual cortex, we are bringing the activity of the neuron to a suppressed sort of state, right? So because there is no input anymore coming. And so the neurons sense that there is less and less and less activity. And so suddenly understands that what he has to do is to bring this activity back to a dynamic range. And so does perform what we call upscaling. So what you expect under this condition to see an increase, basically, of a synaptic response. And so this is exactly what happens. So what you do is basically record from the visual cortex miniature excitatory postsynaptic currents from the control hemisphere, so ipsilateral to the eye that were injected TTX, and from the experimental hemisphere, so contralateral to the eye where we injected the TTX. And so what you see indeed is that in the eye, so in the contralateral hemisphere, so what we call here TTX hemisphere, we see an increase of the amplitude, so the scaling up of the synapses as you would expect. And this has been described by many before. And so those are control animal, wild-type animals of the two different genotypes. So we always use litter mates for all our experiments. And so in both backgrounds, I mean, at the end are just black 6 backgrounds, but we see the same. And this is actually completely abolished by mutations in the two genes that we were looking into in mice. So to next see what this is, so basically what we're seeing here is that mutations in different histone modifiers are related to synaptic plasticity defects, and this is not just about Hebbian plasticity, but also about homeostatic plasticity. So in order to understand whether this is something that it's just affecting mice or also eventually true for human cortical circuits, what we did is we turned to organoids. And again, I mean, those are just basically three-dimensional structure that we prepare starting from human pluripotent stem cells or embryonic stem cells, carry mutations in the genes that we want to study. And we let them practically self-organize in a medium that is inducing neuronal tissues, and we'll let them self-organize to form those organoids or server organoids. And so once those become relatively old, we also push them a little bit to get a bit more mature. What we do is we expose them or not to TTX to resemble these homeostatic plasticity experiments that we did in mice. And then we cut, so with these slices of the organoids and measure excitatory postsynaptic currents, similar to what we have done in the visual cortex to understand whether those organoids are able to undergo homeostatic plasticity. And so since in this case, we were the first one doing this because homeostatic plasticity in several organoids, I mean, in general, patch clamping organoids is something that is not really done much. So we first show, in fact, that also in those human-derived, cell-derived organoid structures, we can induce upscaling of the synapses. So again, we apply TTX. The cells kind of detect that the activity is going down, so brings back to the synapses to a dynamic range. So that's why scaling up. So this works well in wild type, and this is in fact completely gone in all the genotypes related to histone modifiers, but not other genes that we have looked at. So indeed, we can really prove in this way that what we really see, and we were really surprised, in fact, about this. So we did all the experiments, I mean, experiments in blinds, and so we really went through a number of different controls. We see that the plasticity is completely abolished. And here, I just want to mention that what I find remarkable, in fact, is that this seems to be a binary type of process, right? So it's a yes or no, so 1 or 0. So it's not that we see kind of intermediate phenotype where we see a little bit less of upscaling. Is either happening or is not happening. And this was surprising and probably will tell us a lot about the biology. It does tell us a lot about the biology. Good. So let's go back now to the part. So I mentioned, right, that I'm a bit upset. We are always looking at the synapse, but we are often not looking at what happens in the genome. And then I showed you a lot of stuff about the synapse, and I'm not showing you anything about the genome. So what happens there? So in order to answer to this question, what we did is a number of different things, and I'm not going to show you all of that because I was afraid that eventually you would get too bored. In general, what we did is repeating the behavioral type of trainings or TTX injections that we did for studying the electrophysiology to induce plasticity. And in this specific case, what I'm showing you is the fear conditioning. So we did train the animals in the same chamber in the same way as when we perform context fear conditioning. But in this case, instead of waiting 24 hours after the training, we sacrifice the animal 1 hour and 3 hours after. So basically, we induce plasticity and we kill the animal and we extract, we dissected the hippocampi, we extract RNA, prepare the library of RNA or of DNA, because we also did ADAX sequencing, and perform RNA sequencing or ADAX sequencing. ADAX sequencing, for those of you that don't know, is just a way to map the chromatin to see what are the open and the closed regions. And here I'm just showing you practically one data set, but because I think at the end this is the most indicative. So what we see is that those are just control animals, so plus animals, littermates of our mutants. And here I plotted a number of differentially expressed genes in the 1 hour after, so 1 hour after, so 1 hour versus on-cage animals, or 3 hours versus 1 hour. So what we see is that the number of genes, so first of all, we see that the training of the animal induced some sort of response. So something is happening when we are training those animals behaviorally, something is happening in the hippocampus that is one can measure at the transcriptional level. Second, we see that in the very first phase, we see, here red are upregulated genes, and blue, I always actually, one should be careful. It's not upregulated genes. It's more abundantly present mRNAs, because we are not looking really at the gene expression. I mean, we are looking at transcripts. And so more abundant, upregulated transcripts are characterized in the first phase. So after 1 hour of the after the training, we see an upregulation of a number of transcripts and very little repression, downregulation. 3 hours after instead, so we see a really huge response, and this includes both up and down regulated transcripts, but we see a little bit more downregulation than upregulation. So just to say, this is a dynamic, and of course, there is already quite a number of work about that, and so I'm not going to go in detail. What happens in our mutants? So what we see there is a completely different and massive different regulation. And in practice, what we see is that there is practically an attenuated response to the induction of the plasticity. So you see that there is, despite the ratio between up and down is kind of maintained. So you see that is always the same kind of here, more repression that activation. There are many fewer genes in our mutants. And this is not just true at the level of the RNAs. So if I would change this with differentially accessible regions at the level of the DNA, I would see the same, or we have seen the same. So in fact, there is a high correlation between the number of transcripts and so how much upregulated were the transcripts and how open was the chromatin. So why is that? Well, what we noticed is actually that despite in the mutants, many of the genes will not make it to the cutoff for being considered differentially expressed genes. Eventually, they kind of had a threat. So the response was just really attenuated. And then, again, I'm not going to show you all this data, but practically what we found is that why in the wild type, the inputs, or in this case, the training with the context field conditioning chamber leads to a precise sort of response. So a very kind of clear gene expression changes. In the mutants, they're kind of a bit more here and there. So there is more variability, and the fold change in general much more attenuated. And this is exactly what we know, actually, epigenetic factors are doing. Epigenetic factors are making this variability. So we know that even during development, our brain is relatively variable. We learned before also about variability across regions, but so usually those factors, basically, they are making those responses more precise. So another way basically to see this is that we think that in wild type, we have a precise and efficient response to the induction of plasticity, while in mutants, we have a variable and inefficient response. So good, basically, I went through how we used all those mutants. I didn't show you the data for this one. It's in fact already published, and for this one is kind of ongoing. But I show you the data for everything else. What I did not show you is how we are using those stop animals. So those stop animals basically are animals where we introduce a stop cassette after the first open reading frame that is common to all the isoforms. And this stop cassette can be removed at will. So basically, we are generating knockout animals. And then when we're injecting them with something that is tamoxifen, we can revert them to become wild type. And so that's exactly what we did. So basically, we generated this KDM6B animal model. This has half of the amount of KDM6B protein. So it's a heterozygous knockout. It behaves exactly the same. We crossed this with a mouse that carry the Cre recombinase, so the ER Cre recombinase, that is a form of Cre recombinase that is released just upon tamoxifen injections. And so then we injected, so we get this mutant animals. So those are stop animals that when I'm injecting, will become then wild type animals. So we then basically inject tamoxifen. And so that's what happens is like, this is the stop animal with a vehicle injection. This is with tamoxifen. We can rescue, even though not 100%, but we can rescue quite well the amount of proteins that is produced. So we're making from a knockout, from a heterozygous knockout to a wild type. So what happens to the phenotypes? Well, we actually see that this injection has a remarkable effect in the LTP formation. And here I'm just showing you LTP as a readout. So here are the stop animals, which as the heterozygous knockout animals show no LTP, so no long-term potentiation. And upon tamoxifen injection, you see that we could rescue the phenotype. And if I would overlay the wild type with the mutants, at this point, it would really exactly overlay. So what it says basically, this experiment, that we can reverse this phenotype. Maybe not everything, and we don't know how much we can reverse, but at least that the plasticity phenotype can be reversed. And so this also in part tells us that this is not just a developmental effect. And then knowing this, what we can do is then use our organoids to screen now for molecules that eventually would also reverse the phenotype in a gene-specific manner, because eventually we still need drugs. And so that's what we did. So we looked at what KDM6B does molecularly, also based on the knowledge that we acquire from the RNA and other sequencing. And we identified some molecules, and here I'm just showing one, that eventually could have an effect in modulating the chromatin accessibility in the absence of KDM6B. And so when we apply this molecule, so this is the homeostatic plasticity, when we apply this molecule in mutant organoids, we see that we can rescue completely the phenotypes, and the same molecule cannot rescue the phenotype in H1L, so the homeostatic plasticity phenotype. So really showing that we can go from kind of understanding a little bit the function of those genes, and then use both mouse and organoid models to identify molecules that we can use to screen, to rescue phenotypes. So let me summarize this, and this is summary 1, but afterwards just a few slides more. So basically what I have shown to you is that we were interested in the communication between genomic plasticity and synaptic plasticity. And so in order to do that, we analyzed the function or the dysfunction, I should say, of 4 different chromatin modifiers or histone modifiers. And what we found is that indeed, deleting those genes from the genome, or one copy of those, leads to a problem in a less efficient sort of communication and between the synapses and the genome, and to a less efficient response during the synaptic plasticity. So now I just want to bring a few more slides. So if you haven't heard any or listened to anything because you are sleeping, you are still after lunch, that's fine. Reset. Just listen the next 5 slides because it's a different story. So you're still able to do one question. So, and this comes to the fact that genetic factors are studied, right? And those are, for us, are always a good entry point because they're easy and accessible. With the resources that we have now, we can do a lot of sequencing, identify genetic factors, and study them in sort of details as I was showing to you. But then there is still this black box, which are the environmental factors. And there, I would argue that we don't have not even nearly the power yet to really understand what those environmental factors are. If we want to go statistically powered studies, we can really not do that yet to understand how those environmental factors could influence brain development. But here I would argue that eventually we can learn something still from genetic factors about potential environmental cues that are important for the brain. And so I will show you in a moment what I mean. So this next few slides is about metabolism and the brain. And again, when I say just metabolism, everyone shut down. Don't do this. I promise I'm not going to show you, or maybe just one thing of the TCA cycle, but no, mostly not. But so what did we do? Well, a few years ago, we identified those 2 families. Again, very rare genetic conditions. We have a few more, but still very rare. That have, this one is for a geneticist is a dream. It's a huge family and has a few children affected by autism and a number of other features. But many also individuals that are unaffected. And then this family was much smaller. And so what we found is that those individuals here in red all had, besides having the disorder, had mutation in this gene that is called SLC75. And I'll tell you in a second what this gene is doing, but practically the mutations that we identified in those individuals were all affecting, so there were 2 affecting very well conserved amino acids of this gene, or the proteins are encoded by this gene. So really saying that the mutations that we found here, or the variations that we found here, are probably mutations as this conservation you see usually on something that is very important. So what is SLC75 doing? So this is this gene is encoding a transporter that is present at any but is present at plasma membranes and transports non-neurotransmitter amino acids. So not glutamate and GABA, but other, just very simple molecules. So isoleucine, leucine, phenylalanine, methionine. So now there are 2 things that you need to know about those amino acids. And so most of them are essential, meaning that there is no a single cell in your body that can produce them. So you are fully, completely, 100% dependent on your diet, which usually is not a problem because we kind of ingest like 100 times more of those amino acids than what we need, but still you are dependent on that. And they are metabolically relevant, so they're implicated in metabolism. And so when we started working on this, actually already a few years after working on this, we actually went back to a kind of very basic questions. So such as, what is the level of essential amino acids in the brain? You would imagine, amino acids are making proteins. So we don't know the amino acid level in the brain. No, we don't know. We don't know in the human, but we didn't even know in the mouse, not much. Does their level change over time? Also not known. And in fact, how does the level of many other metabolites change across development? So we have some information here and there about metabolites, but really not a perfect assessment. And for a perfect assessment, we will have to wait more. I'm not going to show you this, but just an initial assessment would be helpful. So what we did here is taking the cerebral cortex of mice, and mice are not humans, right? It's for many aspects, so it's important to keep in mind. But still, of mice at 3 different developmental stages, E14.5, P2, and P40. This is nice because it kind of, so one side, the animals have employed at those 3 different time points, 3 different feeding strategies, right? So inside the belly, lactation, and normally and independently provide food. But also because the cerebral cortex is sort of enriched a little bit more for different type of neurons. And so what we did is analyze about 500 metabolites, the level of 500 metabolites of mice, wild-type mice at those 3 different stages. What we found is that indeed, you can just do a principal component analysis. You see that the different stages, they're separated very, very well. So practically what I'm saying here, it probably sounds not as a surprise, and it shouldn't be a surprise, but no one has done it really so far, that metabolism is not just one metabolism, but in fact that just based on the metabolites, you can separate mice by the cortex of the mice by age. Those programs are kind of very conserved, if you want. Like here, there are 4 different animals of the same age, and then the next age, and then the next age. You see that actually it's quite reproducible what we see from animal to animal, even if the animals comes from different litters. So really, there must be a meaning behind it. And so to understand a little bit more about that, what we did is clustering the different 400 different metabolites in clusters based on their progression over time. And we came up with 6 different clusters. So, and each of these eventually contains a number of these different metabolites, but to understand whether there is a trend. So some metabolites that kind of change together. We perform an enrichment analysis, and I'm just going to show 3, and really I have 2 more slides. So what we did this enrichment, and what we found is that among the metabolites that are increasing over time, we have metabolites that are related to neuroactive molecules. That's not a surprise, right? So we have a brain that is developing, more neurons coming, more neuron activity. So things like glutamate and GABA, so a lot of metabolites related to that. So this is not a surprise, but it's helpful as a positive control. What was a bit more surprising is to find that among the metabolites that are actually quickly decreasing immediately after birth, we find metabolites related to valine, leucine, and isoleucine. And for us, what was interesting is that those are the substrate of the transporter that we found mutated in patients before, right? And then a few other changes. I'm not going to go into that. So in order to ask what is the meaning of this metabolic program, right? So for instance, of this, what are the consequences of impacting this decrease of those metabolites at this stage? By the way, here, decrease doesn't mean eventually low transport, but could be a higher utilization, so we cannot distinguish. What we did is blocking the access of those metabolites into the neurons and ask what happens to the neurons if you do that. So basically, if we are impacting this trajectory by blocking those metabolites, what happens? And so that's what we did. We deleted the transporter, S-acetaminotype transporter in neurons only. And we asked what happens. Does this have any influence on the neuronal properties? I mean, leave it alone metabolism. And again, I don't have here the time to show you, but eventually this could have an impact in metabolism. What about neuronal properties? Well, actually, it comes that, in fact, has a very important consequences. So if we are removing the gene from neurons, we see a real a significant decrease in neuronal excitability, as you can see here by patch clamp. This is the cortex. We're injecting current, the neurons, mutant neurons are responding much less. Now, I mentioned that those amino acids will go deeply down during, immediately after birth, but then in fact, they kind of stay steady. And in fact, what we find is that these phenotypes observed just early after birth, but afterwards it's gone. And I don't have the time to show you all the consequences this has and how this is linked to metabolism, but just believe me in here that basically changing the level of those amino acids as an impact on the metabolism, and this impacts the neuronal activity. So just practically all what I show you is in this slide here, is that we are studying autism spectrum disorders, and we are studying genes. And by studying those genes, eventually we try to identify factors that are modifying brain development, brain functions, and I show two of such factors that can also be imposed externally, that is, or change from externally, that are epigenetics and metabolism. So how those two would eventually make the ball roll in a different way. So with that, I want to thank my lab. The work that I've shown to you is mostly the work of Bernadette, mostly Bernadette, really, with the help of Matteo, Christoph, and Jessica. And the work on the metabolism is actually spearheaded by Lisa, and of course, funding agency. And thank you.